Okay, it's now my pleasure to introduce the second keynote speaker, MARC WEISSKOPF, who is professor at the Harvard Chan School of Public Health. So Mark, the floor is yours.
Thank you. I guess you're taking care of this. I'm wearing this thing. So thank you to the organizers and everybody that pulled off this meeting and the meeting to come for the rest of the day. It's been phenomenal. I appreciate the invitation to get to talk to you today. Following that tremendous talk about air pollution in India, we're going to dive a little bit more maybe into biology and how that might influence how we think about our epidemiology or where we might go with it. So my talk's on air pollution in the brain. Let me start though by saying that the theme of this meeting, right, is on the sort of recognizing Hippocrates and on airs, waters, and places, and the history and future of environmental epidemiology. So I sought this out and sort of perused it a little bit for what the history of air pollution in the brain was, 400 BC or whenever Hippocrates was writing, and he was actually quite prescient for the brain. He's got things like in a city that's exposed to hot winds, that was the air pollution of the day, I guess, between the wintry rising and the wintry setting of the sun. So he's got things about when inhabitants pass the 50th year, these defluxions, then render them paralytic when exposed to strokes of the sun or the cold, but those in the opposite exposure, namely to cold winds, inhabitants' heads are sound and hard, and they're liable to burstings, and there's some interpretations that might be blood vessels bursting in the brain. And then, of course, in other cities that are exposed to winds between these, the inhabitants have clear voices and in temper and intellect are superior to those which are exposed to the north. So, he had a lot of prescient things in there. I mean, the top is aging, effects of aging. When we get older, these things are going to get worse, unfortunately for us. He's got things about the possibilities of air pollution affecting cardiovascular events in the brain, which is relevant for cognitive function and other neurological conditions. And then down here, he actually mentions the intellect itself. I don't know about the voices, but whatever. You guys can decide which city you're in and which exposures you have and what you're going to be experiencing as you move forward. But fast forward now to where have we come and how did we get started in the current era of air pollution in the brain. One of the earlier things that sort of kicked this off was studies by Lilian Calderon-Castellanos in Mexico City, looking at dogs from Mexico City and in part in comparison with dogs from other cities that were cleaner, and finding in the brains of these dogs some rather dramatic neuropathological findings of things like reactive astrocytosis, degenerating neurons, tangles, ApoE, tau-positive neurons, and all around the brain in cells and white matter activated microglia and vascular effects like blood-brain barrier problems, all things that clearly we don't want to be happening in the brain and seem to be more common in these dogs that live in the more polluted city of parts of Mexico City. And they, as many of you might recognize, a lot of these neuropathologies that she was identifying in the brain are very relevant for neurodegenerative conditions. And so it raises this issue of, is air pollution one of the contributors towards things like dementia? And that's a critically important question because dementia, as I'm sure you're all aware, is a massive, massive problem in the world, right? These are some estimates, courtesy of a former doctoral student, Melinda Power, putting these together, of estimates of Alzheimer's disease or dementia cases across the world globally. And this was the situation in 1950 where each red dot represents 100,000 cases of Alzheimer's disease. And by 2000, that's jumping up. It starts to get more populated with red dots, and by 2050, it starts to get pretty severe, and we're up to estimates of 115 million cases of Alzheimer's disease. So this is tremendously important. This is just dementia is perhaps the tip of the iceberg. There's also cognitive functions before you get to clinical dementia, but this, in knowledge economies as we go, this is going to be massively important. And if there's a contribution from something modifiable like air pollution, we better understand it so that we can potentially actually do something about it. So the epidemiology, that was back in 2002, and it took a little while, I guess, not that long as far as epidemiology goes, to start getting the epidemiology going, but some of the earlier findings when people started to look into this, one of the first was in Germany, just next door, by Randt et al., looking at dementia or cognitive function scores in relation to how close someone lived to busy roadways. And this, I think that's the, which is my, it's not working. Oh, it's a separate one. Oh, look at that. Okay, perfect. So This is getting closer to the busy roadways, further away from the busy roadways, and you see cognitive scores. This is in sort of Alzheimer's-related cognitive performance, decreasing as among those people who live closer to busy roadways. J.C. Chen and Joel Schwartz in the same year looked at NHANES data in the United States of adults. This was in women, elderly women. This was adults 20 to 59. A lot of numbers here, but basically they didn't see any association with PM10, but with ozone, and this was county-wide annual estimates of ozone, scores on tests related to things like attention and memory and learning and memory got worse with higher levels of ozone in the counties where they were residing. We've done some work, we followed along on this in the normative aging study, a group of elderly, population cohort of elderly men in the Boston area, followed since the 60s that we looked at. Now more sort of detailed spatiotemporal maps of different exposures, in this case black carbon, looking at their performance on cognitive tests relative to, again, where they lived, and found, of course, similar findings of lower performance on the mini mental state examination, which is an Alzheimer's screening type test, as well as sort of a global set of tests that they were doing for cognitive function, getting worse with higher residential address tagged levels of black carbon in the Boston area. Now, it's gone on to sort of somewhat explode. I mean, it's growing literature on this issue of air pollution and cognitive function dementia, and I've actually just put up here some reviews that are going over all the papers. This was, kind of quick and dirty. I did this quickly, and so maybe I left out some papers, but I think the general form is correct that, we're seeing in the early days things sort of trickling along, but it's really starting to pick up with a lot more work into these types of issues in the literature overall. And if we step back a little bit and say, well, okay, does this make sense biologically? Well, there's a long history, much of it played out in these halls of ISEE over many, many years, of effects of air pollution on respiratory phenomena and cardiovascular phenomena, getting in through the lungs and getting into the circulatory system, and ultimately perhaps affecting the brain directly or through activation of inflammatory responses. And those cardiovascular effects are very well established. Many of you have done the work to set that up. And we know that cardiovascular effects are going to impinge ultimately on cognitive performance. So it makes sense in this biology that air pollution should potentially play a role in cognitive processes. But there's additional things to think about, and more sort of intriguing and exciting newer work is suggesting that that's not perhaps the only way that these things can be going on. There's certainly the cardiovascular effects from systemic system up through the brain, because it's a very energy-dependent organ. But what we're recognizing now more and more is that there may be another route for air pollutants into the brain, and that is actually directly through the olfactory nerve. So you actually have part of your brain practically sticking out into the environment in your nose. And so as these things are coming up, maybe they don't actually have to go down to the respiratory system and in through circulation. And in fact, that particles can go potentially directly through the olfactory cells and receptors through the olfactory nerve and get back into the brain. And there's more evidence that this now can occur, particularly importantly with things like ultrafines, where they have different properties in terms of how they pass membranes and can deliver materials to the brain perhaps even more efficiently. Some work, animal work by Oberdorster and colleagues have shown this, that particles can get in through the nose and get into the brain. And I just put down here just so that you know, there's also, this is actually a review paper down there on actual effects of pollutants, air pollution, and olfactory function. So one might expect that if this is one of the roots. And I should also point out that actually olfactory dysfunction is one of the very early signs in many neurological conditions, definitely Alzheimer's disease, and some people think for Parkinson's it's one of the first things that you wind up seeing. So that's an exciting, sort of interesting new route by which this stuff can get into the brain, but if we think about the biology and the way this happens, it might also inform kind of what we see epidemiologically. The area where the olfactory nerve projects in the brain, and sort of the most direct place where these things are then going to get to, is right into temporal areas of the brain, medial temporal lobe, which is the sort of hotspot and center for dementia and Alzheimer's disease, and learning and memory and these kinds of functions. So on that scale, it makes a lot of sense that this route might be important. But if we think also a little bit more about these areas, it's also kind of the non-cortical lower, what we call the limbic areas of the brain, that also include structures such as the amygdala, which is critically important in emotional regulation in the brain. So at one point I was thinking, well, if this is the case and this is where it's coming, then and frankly, the biology that underlies sort of learning and memory type tasks are very similar to things that regulate emotion, that we ought to be thinking about mental health disorders and air pollution. This is a huge, huge public health problem that I think does not get enough attention from the environmental perspective. This is estimates from the World Bank Group of people with depression and anxiety going up from 416 million, and this is just anxiety and depression, from 416 million in 1990 to 615 million in 2013. Estimates from the US have that almost a quarter of US people will experience anxiety or depression within the past year, and almost half over their lifetime. Right? So this is a phenomenal public health problem. Global estimates of disability-adjusted life years, we see the classic players up at the top of cardiovascular and circulatory diseases, diarrhea, lower respiratory infection, etc. But neuropsychiatric disorders, large portion of which are the mental and behavioral ones, and then there's the sort of more classic clinical neurological, are third, right up there, and very close to the others. I mean, this is a major, major global public health problem. And if air pollution is contributing to this, we better know it and understand it so we can do something about it. So we set out to sort of examine this now epidemiologically and start this quest of understanding whether it's also related to mental health functions. And we did some early work in the Nurses' Health Study, looking at roughly 70,000 women in that study who had responded to mental health questionnaires, in particular the Crown-CRISP index, which is just one of many possible ways of assessing anxiety, and related that to the spatiotemporal maps that have been used in the Nurses' Health Study for a long time, to levels of PM 2.5 and then the 2.5 to 10 fraction of air pollution, and found not really much signal with the coarser fraction of particulate matter. But for PM 2.5, we were finding that the odds of being sort of a high anxiety person, cut off on that scale of the Crown-CRISP index, was significantly elevated, whether we were averaging over the month prior to the point where they answered the questionnaire, the prior year, or in fact the 15 years of data that we had prior to the first questionnaire being asked. Those are all separate models. I will say, and this I get sort of into the wonkiness of epidemiology methods and stuff. So this is actually saying, well, which of those windows matters? And we also tried to look at what happens if you put the short-term one, the 1-month average, in the same model with the other ones, like the 12-month average. And it certainly looked like that shorter, more recent exposure was more relevant for the anxiety symptoms they were experiencing. And I'll come back to this a little bit because this touches on a epi-methods issue that I think is very important and potentially often very useful in air pollution, of looking at different windows of exposure in the context of something known as negative control exposures to help you deal with issues, or at least understand issues of confounding in the data that you're looking at. This field also has started to burgeon, and this is another similar sort of probably not complete assessment of papers that are looking at these issues, but it's also definitely growing. These are several reviews that have looked at air pollution and mental health disorders, so that this also is starting to pick up in interest. Of course, this is all sort of from questionnaire-based types of assessments. Brain imaging is a very important means by which we can sort of get deeper into the brain and understand maybe a little bit more of the underlying biology that's being affected. This is a paper by J.C. Chen's group at USC looking at just volumetric changes. So that is in the gray matter, the neurons, the sort of size, how much brain matter do you have there, in particular the neurons themselves, in relation to PM in this case. And the sort of pseudo color is showing decreases in cortical brain volume in different regions related to that PM 2.5. And this is, of course, a great way of trying to get deeper into this and see what parts of the brain are being affected, which cells are being affected. You can look at things like the neurons themselves, the white matter tracts, which are the connections between the neurons. You can look at blood flow to see which parts of the brain get active during different tasks, how a task is being sort of carried out in the brain. So it's really a wonderful way of trying to do this, and there's a lot more work that's burgeoning in this area as well, and I think we're going to see more and more of that come, and it's a great way to begin to get at underlying biology. It still gives you, as far as the biology is concerned, is a sort of grosser picture of where things are happening in the brain in a good way, but I wanted to try and step down a little bit and say, okay, well, what is if we start getting to the individual cells and the mechanisms by which these kinds of changes might happen. Now, let me first say that there are lots of processes that can go on in the brain that can be deleterious to different aspects of the brain. Lots of things can happen regardless of whether it's coming in through the nose, the systemic system. You can have direct transport, you can have effects on the blood-brain barrier, astrocytosis, microglial activation, direct neuronal effects of some metals or things of that sort. So there's lots that can happen, and these ones that are boxed in red are all things that we actually see in brains of people who have things like dementias or psychiatric conditions. So there's a lot that could potentially happen. But what I'd like to do for the moment is focus on one aspect of this that I think, but I'm willing to have discussions and perhaps be changed in my opinions, but right now I'm very intrigued by the microglia. And I sort of think that they are perhaps the sort of frontline player in this issue of air pollution in the brain. They're obviously the things that react and try and deal with inflammation and problems that are occurring, insults to the brain. And there's a lot of things I think we can learn if we pay attention to what's happening with the microglia. The first thing is obviously they're there for a reason to sort of protect the brain and help in cases where there are insults. But you can get to problems. If the microglia get activated for too long, they can become, frankly, toxic, and they sort of are overacting and can lead to neurodegeneration and neural problems just in and of themselves. But that activation can come from particulate matter directly, from cytokines, from adsorbed compounds that are being introduced into the brain. As I say, the problem is if they get toxically activated, we can get problems there. That is interesting because of more and more work understanding what the microglia are actually doing. So, when I first did neuroscience, the dogma was the neurons were where all the action was, and the glial cells were kind of there to sort of just, every other cell, glial and the immune cells, were sort of just there to help the neurons, because they're really what's happening. Well, that's clearly not the case anymore, and we're understanding far more detail what these other cell types do. And the microglia are quite interesting in this. So first of all, if you're in sort of late stage Alzheimer's or dementia, you've got plaques and tangles, the microglia are helping clean things up. They're trying to phagocytose some of the tangles and the plaques that are going on. But there is some thinking and some evidence to the idea that even before you get to this sort of late stage, and perhaps early stage, whether that's clinically showing anything or not is unclear, but the idea that these microglia may actually be phagocytosing part of the neuron. So the synapse itself, right, the place where two neurons touch each other and communicate their signals, the microglia are actually playing a role in breaking those apart and reducing that. And that's sort of the first step towards neurodegeneration, because if you don't want a cell communicating with another cell, and you stop that happening, you're eventually potentially going to kill that cell. And that introduces some interesting stuff. This is work, by the way, that Beth Stevens at Harvard is, in medical school, I believe, has been sort of at the forefront of, one of the main players in this. But it brings up these interesting ideas that if you get toxic activation of microglia, do you get aberrant synapse elimination? Do you start to promote these losses that ultimately lead to dementia? And then it also starts to ask, it raises interesting questions I'll get into more about, well, then what windows of exposure matter if you start activating these cells? And there's an interesting phenomenon with microglia known as priming, where they get set at some point and it will determine how they respond later, which I think is extremely interesting and needs to be explored. And it's certainly known, there is some evidence that microglial changes can far precede neuropsychiatric effects in the brain. And so it's getting into issues that I'm going to come back to in a bit, also about susceptibility related to these microglia. So the other thing that starts to tick off in my brain when I think of microglia potentially phagocytosing these synapses and the way neurons connect with each other relates to important work in neuroscience related to development of the brain. So Roger Sperry, David Hubel, and Torsten Wiesel won the Nobel Prize in Physiology or Medicine in 1981. And Hubel and Wiesel won it for their work on ocular dominance columns in the visual cortex. And that's a phenomenon where cells that are coming from the eye and telling the brain what's going on, early in development, they are sort of over-connected to everything in the cortex. And over the course of development in an activity-dependent way, so part of you going around your environment and a kid seeing things starts to sculpt the cortex so that the cortical cells in the visual area wind up forming these columns, where one column will basically respond to the left eye and the next column responds to the right eye, and that pattern sort of permeates the whole visual cortex. And that process of doing that starts from this over-connected cortex, and is absolutely critically dependent on synapse elimination to create the final pattern, the sort of adult-like, mature pattern of how these cells interconnect. So the process of going from here to here is critical for development. It's known, it sort of sets different regions of the brain have their own critical period over which this happens, and once it's done, that critical period is shut off, and so you don't get these kinds of changes anymore. But the process by which that happens is essentially synapse elimination, right? So there it is, synapse elimination again, and so what's now understood more is, well, first of all, let me just say that actually this is one of the theories of autism, that this process doesn't happen correctly, and that you get sort of aberrant synaptic connections that are not, maybe not in the visual cortex, but in other parts of the brain, where things are just not set up right in how the cells communicate to each other. So that starts to get really interested in neurodevelopment and what's going on, and the microglia, their role in potentially phagocytosing synapses now becomes critically important. And there's much more understanding now in the last 10 or 15 years that the immune system plays an absolutely critical role in neurodevelopment and setting up that final brain structure that's so important. And there's in fact hypotheses in autism, there's the maternal immune activation hypothesis, but hypotheses in general that aberrant expression of these immune signals leads to aberrant development of the brain and problems later in life. And the microglia are central to this, in particular because of their synaptic pruning, or at least that's one of them that I'm most interested in. And again, Beth Stevens and her group has been showing, in fact, in great detail, even sort of particular molecules that are involved, when the microglia come and literally pull apart the synapse and say, that one's not useful, we'll leave this one over here, we create the proper structure of the brain. So it leads to the idea, again, that activation of microglia may be critically important in neurodevelopment. And if we think about microglia a little bit more in neurodevelopment, focusing perhaps a little on autism, although I don't want to say it's just that, but the autism work is quite interesting. So in brains of people who have, brains that have been obtained from people who have autism spectrum disorders, one thing you see is that it's enriched for gene variants in synaptic proteins and synaptic genes. So the genes that are kind of responsible for putting these cells together in a proper way. And we know there's a huge genetic component to autism, and so this seems to be one of the main ways it happens. But intriguingly, they're not enriched in microglial genes of any sort, or things that regulate microglia. However, if you look at gene expression, it seems like the microglial genes are the ones that are different in the brains from children with autism compared to the synaptic genes. The synaptic genes are not changing, the gene expression for microglia are. There's also interesting evidence in animals that early exposures to a variety of things, like stress or diesel particles or inflammation in general, affect microglia number and their function. And Stacy Bilbo at Duke has done a lot of work in this area that's really interesting. She also has looked at things, she's sort of formed this microglial development index based on genes that are being expressed by the microglia, and finds that it's in fact accelerated in human brains, and intriguingly in Alzheimer's brains too, so whatever that means. But that also seems, I don't think I have it here, but more in the autistic, sorry, that is in the autistic brain, but it sort of suggests that this may be a way in which the environment layers on to the genetics of developing autism. But if we don't care about autism specifically, we should just think generally about early brain development. What about air pollution and early brain development? So clearly in animal studies, there can be dramatic effects of exposing animals to ambient particles. This is work by Debbie Corey-Slechta at Rochester and her group, looking at ultrafine exposures to animals and the effects on the brain. This is air exposure at the top, this is concentrated particles in the bottom, and she sees these effects in a graded manner. This is the sort of most extreme versus the controls, and you get very dramatic effects here. This is large ventricles, things of that are pathologies that are similar and in parallel to things that we sort of see in autism brains as well, suggesting that there may be a role there. In the epidemiology now, this has started to expand as well in terms of air pollution and neurodevelopment in general. This is work of ours from Israel with my colleague Raanan Raz, who's at Hebrew University, looking at NO2 exposures, model of NO2 in the Tel Aviv area with over 2,000 children with autism and a bunch of controls, and looking at that NO2 in the postnatal 9 months and the pregnancy period, and seeing increased odds of autism with the postnatal exposures to the traffic pollution that we think NO2 is a marker for. there are various other people who have seen things like this in different settings. You probably are noting here that this pregnancy one looks actually a little bit lower, like it may be protective. We think that has to do with a epidemiological bias known as live birth bias that we also published a paper on talking about, and I'll come back to that as well when we think about issues we have to consider with looking at air pollution and brain effects. But this area too has seen burgeoning work, and again, similar about the publications over the years have been growing, and this is not just autism, we're looking at neurodevelopment effects in general of air pollutants. There's several reviews up here also that have gone over this topic. So another area that's clearly growing and hopefully will continue to grow. So if we think though about that microglial issue again, and are there other ways that it might inform our epidemiology? Other interesting things about that microglial work is that in those early animal exposures I talked about that affect microglial number and function, it seems like male animals are much more sensitive to that effect. And now autism certainly, and some neurodevelopmental disorders, tend to be very heavily male dominated. That microglial development index also shows sex differences. And so it might suggest that these microglial effects, which may be again the ones that are more related to the environmental exposures, have male biases. And certainly in our epidemiology of pollutants and autism, we definitely see this. And others, it's not always looked at in this way, but there certainly are others who see things sort of like this, where we see the most dramatic effects of these in the boys and not in the girls. Now, of course, it's dominated by boys and less girls, but still, it seems like a lot of these air pollution effects may be more dramatic in the boys. So, getting back to Hippocrates, or as in deference to Marianne, he would be very upset if I didn't actually say Hippocrates, which is the correct pronunciation. Where are we now in all of this? I mean, there's growing evidence for adverse effects of air pollution on a variety of brain disorders. So clearly this is growing, whether that's neurodevelopment, autism, neurodevelopment in general, autism specifically, or there's studies on ADHD as well, or whether it's in the elderly and cognitive function or dementia. And I didn't even talk about a lot of work on other neurological phenomena. There's a lot of work around Parkinson's disease with the evidence being perhaps a little bit more uncertain. There's very recent work on amyotrophic lateral sclerosis. There was a very small study, I think first shown here at an ISEE meeting out of Pittsburgh, but Roel Vermeulen and his group right here in Utrecht have been looking at this in the Netherlands and seeing associations. We, with Aisha Dickerson and Johnny Hansen at the Danish Cancer Society, have been looking at this in Denmark and seeing associations with occupational diesel exposure in ALS. So there are other places where we think this might appear. But as we also think about that, we need to think about a variety of issues that are relevant for our study of this. The first is heterogeneous outcomes. So all these brain disorders really are not that clean always, right? So autism obviously is an autism spectrum. There's a huge variety in the phenotype you see in autism, and that's true of general neurodevelopment and the sort of cognitive profile and behavioral profile of kids as they're growing. That's quite varied. Same with the elderly, the sort of the specific cognitive profile that you see start to decline can differ widely. And does this mean that different exposures, different susceptibilities create different effects in these groups? Uncertain, but something we need to be aware of. There's also the issue of just how to categorize dimensions, right? So this is quite problematic. And the classic thinking, for example, is that there's a vascular dementia and there's an Alzheimer's dementia, and sometimes, do we want to look at risk factors for one and risk factors for the other? But the field is certainly moving towards the idea that they're not necessarily different. There's a lot of overlap, and should we be making those distinctions in dementia? And my other colleague, Jennifer Weave, would be very upset if I didn't point out that dementia is an incredibly heterogeneous pathology, right? So this is an example. If you take a bunch of people who are clinically diagnosed with dementia, what's the pathology you see in the brain at autopsy? Well, first of all, there's actually a non-zero percentage of these folks who don't show any pathology as far as we can tell. A healthy number of them, of course, show an Alzheimer's disease pathology. A number have sort of vascular pathologies in there, or Parkinson's and Lewy body pathologies. But the majority of these are just mixed, right? You're seeing all sorts of different pathologies in the brain. So that's problematic from an epidemiological perspective when we're trying to figure out what our outcome is. If you take people not clinically diagnosed with dementia, this is also a problem because, of course, the majority of those don't have any pathology in the brain, but many of them show Alzheimer's-like pathology, vascular pathology, and others. So this creates problems as we're trying to look at it outside of the brain in an epidemiological context. I can't do this without talking about epidemiological issues in these kinds of studies, a main interest of mine. We also have to consider effects when we're conducting our epi studies about things like selection biases, hugely important both if you're studying aging or children. Particularly if the exposure of interest you're looking at is related to both the outcome and the people getting into your study, that is a classic problem with cognitive declines or ill health in the elderly, but also in the children. We've published a few papers talking about how these biases can affect your results. Typically probably biasing to null, but possibly beyond, and making things look protective when they shouldn't be. There's also the issue, of course, as always, with potential confounding, particularly SES. I mentioned negative controlled exposures as a way to sort of assess that. In cognitive function in the elderly, one issue is that decline seems to be less susceptible to these kinds of effects of SES as just sort of where your state is at any moment. So looking at decline is probably a little bit better, and in any case, that's sort of more relevant for dementia. It's how you are changing over time. There's issues about the exposure metric choice affecting what your confounders might be. And then the other issue is that a lot of studies just typically sort of assume a linear relationship, and we ought to think about other possibilities. So where do we go in the future for this kind of stuff? Well, clearly we need to have a better understanding of what components are the relevant ones. A lot of work has been done on particulates. What about the components of those gases? Ultrafines, I think, are going to be potentially interesting, but we don't have a lot of data on that yet. And that's in part because of limited exposure data, and getting more of that is critical. I think Roel and his team here in Utrecht are doing, in fact, some modeling of that in this area, so we need more efforts like that. And then, of course, other hazardous air pollutants. Brain imaging, clearly a big issue, and we should be doing more of it. Good way to get into the brain. There is actually ways with PET to study microglia, so maybe we should be exploring that. But we should also consider other ways, perhaps lower tech. Brain imaging is quite expensive, hard to do in the field setting. We've been doing some work with the acoustic startle response as a sort of lower tech physiology way of assessing the brain. Anna Maria Mora and Brett Ashkenazi have been doing some stuff with functional near-red spectroscopy, also very much more amenable to using in the field. So I think these are other approaches we need to be considering. And then what about timing? When does exposure matter? And I think here the microglia example can help us as well. First of all, most neurodegenerative studies to date have really considered current exposure, so at the time that they look at the cognitive function or the dementia. And timing issues are looked at more in neurodevelopment in terms of pregnancy or after, but it's still, there's uncertainty there. It's not completely clear. We need to do that more. The microglia seed the brain very early in pregnancy, in fact, before the brain has closed, and then they proliferate into the immediate postnatal period and have a dieback after that. But there are key time periods like early neonatal, when the early synapse formation is occurring, and the juvenile, when it's being pruned back. So we ought to be considering those, and then once it's more or less stable here, that may be a different issue. But we also really need to think about this microglial priming issue, and I think there's potentially some really interesting stuff that might come out of this. There are parallels of this with Parkinson's disease and even mammary cancer, where one of the animal models, or a couple of the animal models of Parkinson's disease, are exposures in utero or early childhood that not only predispose to getting the disease, but make the animal more susceptible to later exposures that would be dopamine toxic or cancer inducing. And I think there may be something, it's possible with microglia, something like this is going on, where early exposures may sort of set the microglial or immune responsivity, and then wherever that is may be determining how you respond to an air pollutant exposure subsequent to that. There was actually a very interesting poster by Hector Olvera, who's now at Portland at OHSU, on early stress experience measured with ACEs affecting your responsivity to an air pollution exposure later in life. So that raises all sorts of interesting epi questions, such as, what are the exposures here that matter? Maybe it's not just air pollution, it could be stress, other toxins, the microbiome that sets up where you will be. And it also says, if you want to look at this connection, maybe if we understood where someone sat as far as this was concerned, we would understand how this is affecting neurotoxicity. So we could go from here at the base as a modifier of the air pollution neurotoxicity effect. And if we see that, then the question is, we ought to be looking also just at that. How do early exposures affect your state, your microglial state, and how you're going to respond to a later exposure? So in some ways, this becomes a sort of another mixtures type problem where there may be a mixture of early exposures that put you at some set point that determine how you're going to react to air pollution later. And the last sort of exciting point about that is that there are thoughts that if microglia are playing an important role, that actually may provide therapeutic or intervention opportunities if we can target things towards the microglia to perhaps modify how someone might respond to air pollution or really any other type of exposure later. So with that, I just want to thank the people who really make my life on a daily basis fun and do a lot of this work. This is my group at Harvard and some former folks that I've worked with as well over the years who have influenced my thinking in this realm greatly. So, thank you.
Thank you very much for this fascinating presentation combining epidemiological and biological evidence, making it all making sense. So with that, I would like to close this session and invite you to the coffee break, and maybe if you want, ask some further questions to Mark and Anna about their fascinating presentations. Thank you very much.